So this is a review of, or a brief review of what we talked about in terms of the different types of interactions that stabilize the different levels of protein structure. So hang tight, we'll go through those. This will be kind of a brief video. All right, so first of all, whenever it comes to protein structure, you have to know that there are basically four different levels. There's the primary, secondary, tertiary, and quaternary structure. Okay, so the primary structure is going to be, well, that is just the peptide bonds. So the amino acid sequence. So those are peptide bonds that determine the structure of the primary sequence, or of a protein. Now the secondary structure, this is characterized by a couple of different local structures, and these are known as alpha helices, beta sheets, and beta turns. Now, when it comes to what controls the structure of these are largely going to be backbone, hydrogen bonds. So how the backbone of a polypeptide interacts with the backbone of a polypeptide is going to dictate whether or not, or basically control the secondary structural elements that are present. Now the tertiary structure, that is going to be side chain interactions, largely. Chain interactions. Now what's great about this is the more that you know, or the better that you know your amino acid R groups, the better you understand the tertiary structure of a polypeptide or of a protein. For instance, the types of interactions that can exist, you have to think about the different residues. So if you think about something like serine, so serine is CH2OH. That's a group that is great for hydrogen bonding with other R groups. If you think about something like aspartic acid, ASP, at a physiological pH, that is going to look like this, CH2COOO-. So that's negatively charged. That is a great candidate for any sort of ionic interactions with another, either something in the solution or another amino acid. Now, when it comes to the tertiary, or the quaternary structure, this is similar to the tertiary structure, however, it is multiple polypeptide chains. Multiple peptides. So how do all these different polypeptides interact with one another? Now, if you think about a single protein, there we go, there's a single protein, a single polypeptide chain. It folds into this neat 3D structure. Now, there are other proteins, for instance, like hemoglobin. Hemoglobin is actually a dimer of dimers. So here would be one subunit, or one dimer of dimers, and then here's the other one. So the 3D shape of a protein like hemoglobin would best be described by looking at the multiple polypeptides, so the quaternary structure of that protein. Now, that is going to be controlled, again, by those side chain interactions. So how do those side chains of one polypeptide interact with the side chains of another polypeptide? How do they contort themselves in space to make an overall 3D structure? So in this lecture, one of the things that we did was we dove a little bit deeper into the secondary structure. Okay, so we looked at alpha helices and beta sheets. Okay, so an alpha helix is, well, it's just that. It's a winding coil that wraps in on itself, and it's going to be stabilized by backbone hydrogen bonding. So here's a bond, here's another bond, and it just kind of continues on in perpetuity. Now, a beta sheet is a little bit different than that. A beta sheet is something that, if you look at this as our N-termini, and it goes on, it wraps back like this, you have backbone interactions between these polypeptide chains. So I'm going to go ahead and draw that, and then there is our C-termini there. Okay, alternatively, you could have something that looks like this. C termini, N termini. We have our backbone interactions between here. Now, what does this mean, our backbone interactions for our beta sheets? What does that mean for our R groups? That means that our R groups are right out here. R1, R2, R3. Keep in mind, I'm not drawing this to scale, really. Let's call it 45. R46, and then R47. These two different beta sheets that are represented here are known as parallel or anti-parallel. This would be a parallel beta sheet, and this would be an anti-parallel. Now, when it comes to organization of these, does this just spontaneously happen? Do these two different structures form without any sort of influence? No, they don't. They actually are influenced by the side chains. So amino acid, or I'll just write AA R groups. So the different R groups do have a bearing on what type of an alpha helix or a beta sheet is going to be formed. And this was largely attributed to two researchers, Chow and Fassman, who wrote a review article in the 1970s, and they scored every amino acid. They gave every amino acid a propensity score. And so that would categorize, or that propensity score on a scale from 0 to 2, they basically ranked every amino acid. Is it likely to form an alpha helix or a beta sheet or something like that? In the lecture that we did on this, we talked about the different scorings, what amino acids would have a preference for an alpha helix or a beta sheet. Go ahead and review that, as well as the PowerPoint presentation that I posted on this. And that's going to be ultimately very helpful when it comes to making these sorts of predictions. Now, I will not ask you to recall propensity scores, but I will ask questions like, based on, is a small amino acid likely to be found in an alpha helix or a beta sheet or something like that. In addition to that, we also talked about, well, the math that goes involved with looking at the size of an alpha helix. How tall is that alpha helix? How many turns does it have? How many complete turns does it have? So make sure that you're familiar with how to do those sorts of things. All right, well, thank you very much for watching. I hope this helped out.